All right, so we'll get started today. Welcome to the Philip J. Curry's virtual speaker series for April. We are very happy to have Joe Moysiuk here with us today. Joe is a PhD student at the University of Toronto and the ROM, studying the Burgess Shale. So he's going to be telling us a little bit about his research today in the Burgess Shale and around early life and the origin of vertebrates and arthropods in particular. If you have questions, feel free to put them in the chat. And after Joe's talk, we'll take a moment to go through all of those questions. So without further ado, I'd like to introduce Joe.
Thanks very much for that introduction, Emily. I'm just going to share my slides here. So yes, I'm very happy to be here today, talking to you a little bit about some very strange fossils from very early in Earth's history. So weird wonders and spaceships, and what these have to do with the origin of the group of animals that we call the arthropods. And hopefully by the end of the talk, I'll have convinced you that there's more to paleontology than just dinosaurs, and that there's a lot of really cool invertebrates that we can learn about from Earth's deep history. And so I'm part of Jean-Bernard Caron's lab at the ROM and at the U of T. And in this lab, we're particularly interested in this phase in Earth's history that's called the Cambrian explosion. And so this is a relatively brief period in Earth's history, a little over 520 million years ago, when we see the origin in the fossil record of essentially all of the major groups of animals, all of the major body plans of animals that we still have with us today. And so we see early ancestors of, for example, the vertebrates, as well as the arthropods, which include modern insects and spiders and crabs, early ancestors of modern segmented worms and jellyfish, and all manner of other animals. And what's kind of interesting about this period of the Cambrian explosion is that we see the origin of these body plans, but then we see the relative stasis of these body plans thereafter. And so the big questions that we're really interested in are, how did these body plans come to be in the first place? And then how have they become so stable over time? And I should say that this pattern that we see with the Cambrian explosion is not just a phenomenon of the fossil record. We also see evidence of it in the genomes of modern organisms. And so there's evidence of very high rates of evolution happening in the Cambrian, as well as these elevated rates of gene gain and loss in many of the early branches in the animal tree of life. And so something interesting seems to have been going on in terms of evolution in this time period. And so to study what was going on here, the group that I've chosen to focus on are the arthropods. And these are astoundingly diverse today. They include all of the animals with segmented bodies and jointed limbs. So like insects, spiders, crabs, and all kinds of less familiar looking organisms like the ones you see here. So these guys make up about 80% or so of modern animal species. So they're incredibly diverse today. And they're also incredibly diverse in the fossil record, including in the Cambrian period. So these are a major evolutionary success story ever since their origin. But how did the arthropod body plan, this ridiculously successful body plan, first evolve? So to get at this, we might be able to get some information by looking at the closest living relatives of arthropods. And those are the velvet worms, or the onychophorans that you see on the left here, and the tardigrades, or the water bears, that you see on the right. And what comes to mind immediately is that there are some major differences between these guys and modern arthropods. So they don't really have a segmented body or jointed limbs in the same sense as modern arthropods, for example. And so in some ways, we're kind of limited by looking only at these modern organisms. And so we might want to turn to the fossil record to get an idea of how these body plans came to be. And so one fossil that's been particularly of interest and significance is this creature called Anomalocaris. So this was first discovered way back when the Burgess Shale was discovered, and even a little bit before in the early 1900s. And it was known from these different body parts that were at first thought to be unrelated that I'm showing on the left here. So we have these strange spiny looking claws. We have these round oral structures, which are lined with teeth. And then we have this strange body with lobes along the side. And so originally these three fossils were thought to belong to a shrimp, a jellyfish, and a sea cucumber. And it wasn't until the 1980s that Harry Whittington and Derek Briggs were able to piece back together the different pieces of the animal by finding more complete fossils and realize what this creature looked like in its entirety. And it's incredibly bizarre looking, as you can see in their reconstruction on the right. There's really nothing that looks like Anomalocaris around today. And so because of this, Whittington and Briggs were sort of reluctant to place Anomalocaris in any known living phylum of animals. And so they said, we do not consider it an arthropod, but a representative of a hitherto unknown phylum. And this was a major inspiration for Stephen Jay Gould's famous book, Wonderful Life, which focused on the Burgess Shale. And Gould essentially was making the argument that we had this rapid radiation of all of these different living body plans, but also extinct body plans back in the Cambrian, and then that these body plans were sort of winnowed out over time. Now, our views have sort of changed a little bit since Gould's time of writing this book, but Anomalocaris is still a real oddball when we think about its morphology. So we now view Anomalocaris as being a distant relative of the modern arthropods. So we call it a stem group arthropod, meaning that it diverged prior to the common ancestor of all of the living arthropods, but after the divergence of the living arthropods from their close relatives, the velvet worms and the water bears. And so because of this particular position on the evolutionary tree of the arthropods, animals like Anomalocaris have this potential to teach us a lot of interesting things about how the arthropod body plan actually came to be and the sorts of character changes that were happening in those lineages moving up towards the arthropods. So over the past few decades, there have been a plethora of discoveries from many Cambrian sites around the world, which have greatly expanded our view of these early arthropods related to Anomalocaris relative to what was known when Harry Whittington and Derek Briggs published their famous paper. And so you can see that we now know this stunning diversity of these different early arthropods. And in some ways, this makes our job of understanding arthropod evolution more challenging, because with all of this diversity, how do we understand the different character changes that actually accrued leading up to the modern arthropods versus all of these little side lineages that popped off along the way? But this is a really fertile ground for new research. And this is what my PhD thesis largely focuses on. And so in particular, I focus in my PhD on this one group of early arthropods, which we call the Radiodonta. That name basically means radial teeth, referring to the mouth parts of these animals. And so this is the group that includes Anomalocaris, as well as some of its close relatives. And if you want to study these radiodonts, the best place, or certainly one of the best places in the world that you can go, is the Burgess Shale in the Rocky Mountains of eastern BC in Canada. So this is this incredible site that's about 506 million years old, and it preserves an astounding diversity of these Cambrian organisms. And the Burgess Shale itself is really important for us for two main reasons. So one of those reasons is that the fossils from the Burgess Shale are incredibly well preserved. So when we think of fossils, we typically think of things like bones and shells and teeth, the hard parts of animals that don't really decay away too readily and can be preserved easily. But at the Burgess Shale, we have exceptional preservation of even things like soft tissues. So we may find preservation of things like the eyes of these animals, or even their brains and nervous tissues, or their digestive tracts still containing their last meals. And so you can imagine that with this type of preservation, we get this incredibly rich window into what life was like at this time in the Cambrian period, which is kind of indicated in this diagram that I'm showing on the right here, where you can see the difference between what the Burgess Shale fossil record actually looks like in the top panel compared to what it would look like in the bottom panel if we didn't get this exceptional preservation of soft tissues. So that we have this really remarkably enhanced window of the Cambrian world. The other main reason that the Burgess Shale is really important is because of its age. So as I said, it's about 506 million years old, and so it dates to the time just after the main pulse of the Cambrian explosion. And so it gives us insight into the fallout of this major radiation of animals. So the Burgess Shale is located just on the border of BC and Alberta. The site was actually originally discovered in 1909 by Charles Walcott from the Smithsonian, and it's located in Yoho National Park. And so lots of research has been done over many decades at this original site called the Walcott Quarry and other sites sort of in the vicinity of that area. But one of the things that is really exciting about some of our work at the Burgess Shale now is that we've been working on these new sites that we found a little bit further south, as I'll talk about in a moment. But I just want to emphasize that there are some key features about the Burgess Shale environment that are interesting to think about in terms of how the organisms were preserved. So we find the Burgess Shale fossils in these beds of shale, which were deposited at the side of this large limestone ridge, which is actually the preserved remains of a submarine escarpment at the edge of the continental shelf. And so it seems like these animals were living around the edge of the shelf, and then mudslides came and trapped them as they slid off the edge of the escarpment and buried them in successive layers at the bottom of the escarpment. And this gives us this sequence of these different animal communities preserved through time. And one of the other interesting things about this undersea escarpment, which we call the Cathedral Escarpment, is that we're actually able to trace it through the Rocky Mountains along a more or less north-south transect. And as we go along, it turns out there are other fossils strewn around wherever this escarpment occurs. So we had mudslides happening all the way along this escarpment. The image in the top right here is just showing you a little bit more about the environment of the Burgess Shale. So at the time, British Columbia would have been very close to the equator, and it would have been submerged under this shallow ocean. And the Burgess Shale was, again, located right near the edge of the shelf, sort of on the margin between the shallower areas and the deep waters. And so, like I said, when I started working at the ROM in 2012, it was a very exciting time to be starting because my supervisor, Jean Bernard, and his crew had just been out at some new sites in Kootenay National Park, about 40 kilometers to the south of the original Walcott Quarry site. And at these new sites near Marble Canyon, they discovered these incredible new communities of Burgess Shale animals. And one of the things that's surprising about these finds is that a very large fraction of the animals that we find in these Marble Canyon communities are actually quite different from the ones that we saw at the original Walcott Quarry. In some cases, they're species that we know of from China, but not from the Walcott Quarry, even though it's only 40 kilometers away. And in some cases, we find completely new to science species. And I'm going to be discussing a couple of those a little bit later. And so I wanted to take you on a little virtual field trip to the Burgess Shale and just show you a little bit about how we actually do our field work there, how we collect the material. And then I'm going to get into talking about how we study the material and what sorts of insights we've gained on some of these new animals that we've discovered. So here's an initial image showing us just preparing to go up to one of these new sites near Marble Canyon. This is our staging ground at the helicopter platform. And you can see that we pack all of our gear into these buckets and bags. And then ultimately, that's going to get packed into these large hanging nets underneath the helicopters so that we can transport all of this stuff up to our campsite. We need these helicopters to help us to get there because many of these sites are very remote and they'd be difficult to access on foot And certainly if you want to spend a month or 6 weeks, as we sometimes spend at the Burgess Shale, you need to bring up quite a lot of gear. And so this is a view of what our campsite looked like at the Marble Canyon site. And you can see it's a very beautiful place to work. We have typically a cook tent for the crew in the middle, and we'll have about 10 people working in the camp at all times. And this is a photo from a smaller camp from an exploratory phase of the field work where we were checking out some new sites. So in this case, we had a smaller crew and we were doing a lot more hiking. And the only place that we could find flat enough to camp here was this snow patch in the bottom of an avalanche gully. And just to give you an idea of the scale of this place, here's the same campsite viewed from a little bit farther away. And so once we've got our camp set up, then the time comes to get down and dirty. And so we start working our way into the outcrops by first uncovering the shale where the creatures are buried with shovels and hand tools, moving away any large boulders that might be situated on top of the shale that's of interest. And then we really get into the shale with various power tools and hand tools like hammers and chisels. And the goal here is to break the shale beds into as fine a pieces as we can possibly split them into. And this is because finding the fossils is a little bit of a game of skill and a little bit of a game of luck. It's a game of skill in order to find the right beds in the first place, the event beds where many of these animals can be found. And then once you actually find those beds, it's a game of luck to be able to crack the shale along the particular plane in which any particular fossil can be found. And so in some of these beds, you can have literally hundreds of animals within like a square foot of shale. And so we really want to be careful to split the shale as finely as possible to try and find as many of these organisms as possible. And once we find the organisms, we actually typically use rock saws to cut them out to make sure that we don't damage them. And we cool the rock saw blades with water. And so we need a lot of water to do our work. So this was our hazing of some of our initial recruits in the 2018 season. We asked them to go and collect snow for the rock saw water supply on their first day in the field. And I think they were a little bit surprised by this task. And so once we are finished with our expedition, we pack up all of our gear and our fossils into these metal cans once again, and we stack them into the helicopter nets and fly them out. And then ultimately they get trucked back to the Royal Ontario Museum in Toronto, where we can study them. And you can see an image of Jean Bernard studying some of these fossils under the microscope in the upper right here. And it's once we get these fossils under the microscope that we really start to see how beautiful they are and how well preserved they are. Many of these things are very difficult to see in the field, but when you get them under the right lighting conditions, and in particular under polarized light, then all of the details really pop out. And you see this stunning quality of preservation. And so I just wanted to highlight a couple of my colleagues who have been also doing a lot of work on these new findings from Marble Canyon ever since it was discovered. So there's Carmen Nanglu, Cedric Aria, and Alejandro Izquierdo Lopez are my lab mates who have been working a lot on various invertebrate groups that you see pictured here. And you may have seen these in some news stories over the past few years as well. But today I'm going to be focusing on one particular group, again, the radiodonts. And so firstly, I want to tell you about this one find that we found particularly exciting. We ended up naming this animal Cambropaster falcatus, but its nickname in the field was the spaceship because we thought it looked a lot like the Millennium Falcon spaceship from Star Wars. Now, when we first started finding this animal in the field, we were turning up these strange shaped carapaces, which you see an example of on the right here, this large Millennium Falcon shaped structure here. And we really didn't know what to make of these things at first because we had never seen anything like this before. And it was only once we started to find more and more specimens of the animal sometimes associated with other body parts like these different head plates, these claws and mouth structures, that we realized that we were dealing with a radiodont, but a very strange radiodont, unlike any that we had seen before. And as it turns out, Cambropaster is very abundant at some of these sites around Marble Canyon. And as you can see on the left here, there are instances of finding dozens of specimens of the animal all on the same bedding plane. So all of these organisms were living together. Just to give you a little view of some of the body parts again, we have this very distinctive spaceship carapace that we see on the right here. We also have another pair of carapaces that would have covered the ventral side of the animal's head, which you see one on the left here. And these are sort of shaped like a paddle with a narrow neck region at the front. We also have these segmented appendages, which are very stubby, but they have these really long comb-like or rake-like outgrowths coming from the inner side of the appendage. And what's pretty striking about these appendages is that when we take a look at the photo in the middle here, which is one of them situated in sort of a frontal view, you can see that the ventral spines really curve inwards. They're not just straight. And we also find these spines on these spines, so these secondary spines as we call them. You can see these, for example, in the lower right image, are strongly hooked, almost like fish hooks. So this is a really fierce looking appendage. We also find the mouth structures of the animal, which we call the oral cone. You can see a couple of examples here. And so this is a structure that's composed of a series of plates around the outside with additional rows of plates lining the inside of the throat, all of them lined with teeth. And so again, a very ferocious looking mouth. And we even find some evidence of preserved remains of the anterior part of the digestive tract inside of this mouth structure too. So really nice fine details. And then finally, we got really lucky with this animal because with many radiodonts, we only find bits and pieces of these animals sort of disarticulated from each other. But with Cambropaster, we actually found complete individuals as well. And so this is one of the most beautiful examples where we see the short, stubby little body of this animal situated beneath that spaceship-shaped carapace. And as it turns out, we see these two very reflective patches under low-angle light on the right here, which line up more or less with the notches in that spaceship-shaped carapace. And as it turns out, these are the eyes of the animal. So it looks like the eyes were actually situated in these notches. And then the rest of the body was this very stubby little structure sticking out at the back here, which was lined along the sides with these pairs of swimming flaps. And so putting it all together, we can come up with a reconstruction of the animal that looks like this. And so this animal, Cambropaster, is actually one of the largest animals that we know of from the communities at Marble Canyon. So it gets up to about a foot long. And so this would have been quite a large predator in the environment. Now, Cambropaster was not the only large radiodont that we found at these new sites around Marble Canyon. We also found this fascinating carapace here, which at first glance resembles Cambropaster, but is significantly larger than most of the specimens of that species. And so as it turns out, we believe it's a new genus and species. We called it Titanokoris gainzy. And the second part of that name is in honor of one of our longtime collaborators, Bob Gaines, who you see on the right here, who's an absolute wizard at finding the best Burgess Shale beds that contain the richest fossil assemblages. And so you can see a couple of the fossils of this animal that we found shown on this slide, including this one remarkable carapace of the animal on the left, which in itself is about a foot long. And so we estimate that the total animal would have been about 2 feet long. And again, this makes it certainly one of the largest animals that we know of from the Cambrian period. We also have some evidence of the frontal appendages and the mouth parts of Titanokoris. Not as well preserved as we saw in Cambropaster, but from what we can see, they appear to be fairly similar. And again, just showing the size of Titanokoris, we can compare that one large carapace to the most famous specimen of Anomalocaris, which is sort of an iconic giant from the Cambrian period. And you can see that Titanokoris is absolutely massive compared to this most complete known Anomalocaris. Although we do actually know that Anomalocaris probably got bigger than this based on isolated body parts. And so putting this together, we can reconstruct the Marble Canyon community, something like this, with our Cambropaster chasing after a school of early fish, and then Titanokoris coming onto the scene as well. So we have these two large predators co-inhabiting this one environment, which we think is really interesting and indicates that there must have been a very rich food source in this community. So the last species that I want to go over is one that's called Stanleycaris herpex. And this one is known from less complete material than the other two. So far, we only know about the appendages and the mouth parts from this animal, although some work that I'm actually working on right now will reveal other body parts of the organism, so stay tuned for that. But just taking a look at the appendages and the mouth parts, for starters, we can see some similarities to the two species I already talked about. So we have these long, curving, rake-like spines coming from the underside of the appendage. But we also see a remarkable array of these other spines, these small hooked outer spines that you see in the image on the right. And then another set of spines, which are sort of trident shaped, that you see in the image on the left. And these ones actually project towards the midline of the animal, sort of perpendicular to the claw. You can see a couple of other examples of the claws on the left here, as well as some examples of the mouth parts of the animal. And so once again, we see this ring of plates that are lined with teeth, which is very characteristic of the Radiodont group. And so we can reconstruct the known body parts of Stanleycaris, something like this. So we have these interesting new species from Marble Canyon and the vicinity around Marble Canyon. But what can we actually say biologically about these organisms? So firstly, I want to talk a little bit about what we know about the feeding modes in radiodonts. So we see in some of the appendages that we've seen already, like Cambropaster and Stanleycaris' appendages, this great diversity of different shapes and sizes in appendage structure. And looking more broadly among some of the other radiodonts that we know from the Burgess Shale and elsewhere, we see even more diversity in terms of the structure of these claws. And so the first thing that we can do is take a look at some modern arthropods and see if we can find some analogs that might tell us a little bit about how these animals might have been feeding. And so, for example, we see appendages that look a little bit similar to what we see in Anomalocaris in some modern arachnids. And so we think that these appendages would have likewise functioned for grasping prey. We also see appendages that look a little bit like this particular radiodont in the center image here, which is called Titanokoris. We see analogs in things like modern krill, which use their appendages for filter feeding. And so we think that some of these radiodonts were actually feeding on microorganisms living in the plankton. And then finally, in the lower center image, we see this very claw-like appendage of a radiodont called Amplectobolua. And this is very similar, again, to these claw-like appendages that we see in some modern arachnids, for example. And so it's pretty clear that these things are used for sort of pinching and tearing and grabbing prey. And so what about more unusual appendages like those of Cambropaster? So in this case, we don't find so much of a clear analog in modern arthropods. These are really strange appendages. The closest thing that we can come up with is that there are these similar sort of rake-like appendages that we see in some modern decapod crustaceans, for example, as well as in some extinct eurypterids or sea scorpions. And in all of these cases, it's thought that these appendages function for raking through the sediment and pulling out any small organisms that might have been living there. And so we think that Cambropaster and Titanokoris were probably doing something similar, raking their appendages through the mud, disturbing the sediment, and then pulling out any organisms that might have been hidden in there. And this also sort of matches up when we think about the whole body form of Cambropaster. So Cambropaster has this really elongate head with the eyes of the animal situated almost close to the midline of the body, and then this really stubby sort of trunk region. And this sort of form is often seen in bottom-dwelling organisms today, so things like horseshoe crabs and true crabs, and we also see it in some extinct organisms, like again, some eurypterids and some trilobites, and even early fish. And so this seems to be characteristic of these organisms that spend a lot of time hanging out near the bottom and feeding on things in the sediment. Now, switching over to thinking about Stanleycaris a little bit, in this animal, we have those same sort of rake-like outgrowths on the inside of the appendage. And so it may well have been doing something similar to Cambropaster and Titanokoris in terms of hanging out near the sediment and feeding on buried organisms. But we also see this really interesting set of these medial trident-like spines that I mentioned before. And what's striking about these is that we see very similar looking structures in the mandibles of some modern arthropods, like these copepod crustaceans that I'm showing on the right here. And so we think that in Stanleycaris, the appendages may have additionally had a sort of mandible-like function where it could have brought these things together in opposition to crush prey items along the midline. And what's kind of cool, a cool little detail about this is that in Stanleycaris, which I'm showing in the upper left images here, we see these very long and sharp trident-like spines along the midline, which are comparable to structures that we see in modern copepods, for example, that feed on soft-bodied prey. Whereas in this other species of radiodont, which I'm showing on the lower left here, which is an animal that's closely related to Stanleycaris, but still remains undescribed, we see these very stubby, stout, and sort of robust-looking spines, which are more comparable to things that we see in shell-crushing crustaceans. And so even among these relatively closely related radiodonts, we think there's some ecological variation going on in terms of the feeding modes that these animals were engaging in and what sort of prey they might have been specializing on. So this is pretty cool. So now, what about the evolution of these different body forms in radiodonts? So Anomalocaris, of course, is the most famous of the radiodonts, but we can see that it's very different from the Cambropaster and Titanokoris that I showed before. So Anomalocaris has these frontally positioned eyes, these very long grasping appendages, and a much longer, more streamlined body. And so one of the things that I'm really interested in is how did these different groups of radiodonts become so differentiated? And how can we reconstruct what the ancestral radiodont might have looked like? And this, by extension, has some significance in terms of thinking about the early evolution of modern arthropods as well. And so here's a simplified tree of the radiodont group and these two major subgroups of radiodonts, which I'm just calling the herdiids and the non-herdiids. And you can see that as a whole, the radiodonts form this sister clade to the modern arthropods. And so, again, by reconstructing what the ancestral radiodont looked like, we might be able to get some insight into the lineage that's leading towards the modern arthropods as well, and what sort of characteristics it might have had. And so just mapping out the different appendage morphologies here along our simplified tree, we can see that we have this sort of dichotomy between the herdiids and the non-herdiids, where the herdiids tend to have these really stout, short appendages with long rake-like outgrowths, and the non-herdiids tend to have these really long appendages with shorter spines, typically adapted for grasping, although sometimes for filter feeding. And so when we map back our morphologies along our tree and try to reconstruct what the ancestral appendage might have looked like, we can come up with something that's a little bit like this. So it's probably a relatively elongate appendage, which was probably adapted for grasping prey. And we don't know as much about what the spine morphology would have been like on this appendage, so there's still a bit more work to be done. Now, thinking about some of the other aspects of the form of these radiodonts, like the head morphology, for example, which is so differentiated between, for example, Anomalocaris and Cambropaster, we still know a lot less about this. So we know that at some point we had to get some sort of a transition either from a short head to a much longer head, or the reverse. And so we're hopeful that some new finds, like for example, the material that I mentioned of Stanleycaris, which we're working on right now, will help us to resolve this problem a little bit more. One of the other things that we've been learning a lot about radiodonts with recent finds is a little bit about how they grew and developed. And so here's a couple of photos based on a nice study from Liu et al. In 2018, where they found a very small juvenile of this one radiodont called Lyrarapax. And we can see that the juvenile specimen looks pretty similar to a much larger specimen of this animal. So there doesn't seem to be a lot of change going on in terms of the growth between these two specimens. However, we know relatively few specimens for most of these species of radiodonts. And in this case, the smallest known specimen here is still about 2 centimeters long. And so we don't really know anything about what was going on in the very earliest stages of development of radiodonts yet. Now, in our work, we were able to do a little bit of growth modeling using some landmark-based work. And so we were able to plot how the form of the carapace of Cambropaster changes as it grows larger. And so in this case, we did actually find that this animal changes a little bit through its development. So the sort of lateral wing-like structures on the carapace tend to spread out and get a bit shorter as the animal grows larger. And we found this kind of interesting for a couple of reasons. And so one of those is that we were able to extrapolate this sort of growth model and figure out what Cambropaster would have looked like if it grew even larger than the largest known specimens. And this is the image that you see on the right here. And so by comparison with the much larger Titanokoris, which I'm showing in the upper right, we can see that there are major differences between the expectation that we have for Cambropaster versus Titanokoris, which is a much larger animal. And so this is one of the reasons why we think that Titanokoris and Cambropaster are actually two different species that are coexisting in the same environment, as opposed to just different developmental stages, with one being larger than the other. Now, one of the other interesting things is that there's another radiodont species from China called Stanleycaris, which in fact looks very similar to Cambropaster in its extra large extrapolated modeled form. And so it's possible that there are some processes relating to the rates and timing of development which are impacting the evolution of radiodont carapace shape. And so this is something that more specimens may help us to clarify exactly what sort of changes in rates and timing may have led to these changes in shape. And then finally, I wanted to talk a little bit about what we now know about the distribution of some of these radiodonts based on various new findings. And so one of the things that I'm really excited about is that since publishing some of these new species from Marble Canyon, very similar organisms have turned up in fossil sites in various other places around the world. And so, for example, in both North and South China, as well as in the United States. And so it looks like Cambropaster and its relatives were actually quite a widespread group of organisms in the Cambrian. And they spanned over, perhaps like 15 million years of time. And so this is pretty neat for a group of animals that was only really discovered and published in 2019. We also know a little bit about the distribution of Stanleycaris based on another study from the United States. And so far, this species seems to be restricted to what is now North America. So it's a little bit more endemic than Cambropaster and its relatives. So we see variation in endemism in these radiodonts as well. And so to finish up my talk, I just want to bring us back to this huge diversity that we see in early arthropods and in the radiodonts in particular. And so we see this diversity in form, we see the diversity in function and ecology, and we see diversity in sort of biogeographic distribution. And we're starting to understand the evolution of this group of animals and how these are not just sort of a weird offshoot of the arthropods that was destined to go extinct, but were their own diverse radiation of organisms, which we're just beginning to understand. And in that respect, the radiodonts can sort of be thought of as a model group for understanding the early evolution of arthropods, because they give us so much insight with these incredibly well-preserved and diverse fossils. And then lastly, I just wanted to say that part of the fun of publishing some of these new finds has been how much interest it's garnered from the public. And in particular, we've had a lot of artists take inspiration from the work, which was really fun to see. So we've had all kinds of beautiful 2D artworks and 3D artworks and paper models and even a Yu-Gi-Oh card come out that are based on some of our work. And so that has been really a pleasure to watch. And I just wanted to include as a final slide here a little promotion for the new Dawn of Life gallery that opened up at the Royal Ontario Museum in December. And so the fossils that I've showed you in the talk today, as well as many, many more, are all on display in this new permanent gallery. And so I encourage you all to come and see it if you can. And so with that, I have many people and funders that I would like to thank. And thank you all for listening today.
Perfect. Well, thank you so much, Joe. It's absolutely fascinating to hear about your research. I've got to say that I'm really jealous of your field sites. They look pretty amazing places to do some work and to live for a couple of weeks a year. I'm sure they have their challenges too, though. So we're just give a few minutes for people to type in their questions if they have them. In the meantime, I want to start with, and so you mentioned that some of these taxa might have been sediment sifters or filter feeders. So my question is, what were they filter feeding on? Like, obviously this was a time before there were things like krill. So what kind of organisms would have there been in the sediment or in the kind of in the water column?
So there is it's a great question, that there is evidence of very small organisms coming from the Cambrian period. We don't typically see them at the Burgess Shale just because the chances of getting a lucky split through these really tiny organisms is pretty low, and observing them in the sort of coarse sediment of the shale. But we do see these organisms preserving at various other sites. So for example, these small shelly fossils, as they're called, are super abundant in a lot of Cambrian sediments. And many of these things are really weird and belong to groups that are no longer with us, although there are also more familiar things like brachiopods, for example. A lot of those things would have been living on or in the sediment. We also see evidence from other exceptionally preserved sites that have different modes of preservation from the Burgess Shale. Some of these really tiny microscopic arthropod larvae in particular. So there's a site called the Orsten from Sweden, which is very famous for this amazing preservation of these tiny, tiny organisms in incredible detail. And so these things are essentially replicated in calcium phosphate and preserved in 3 dimensions. And some of them basically look like you took a modern arthropod and just stuck it under the SEM. And there are also lots of examples of things like small carbonaceous fossils. So sort of like the micro, the micro equivalent of Burgess Shale fossils, which preserve various types of organisms that, again, might have been living in the sediment or in the plankton at that time.
That's really cool. There's a question from Bruce Koshushi. What predators evolved over time to threaten these radiodonts?
It's another interesting question. So as far as we know, these radiodonts were some of the largest organisms that were living in the Cambrian period. And so really the only thing that could have threatened a large radiodont is probably another radiodont. As we get out of the Cambrian period and into the later parts of the Paleozoic, we get other groups of arthropods evolving large sizes, like the eurypterids, the sea scorpions, and we start to get large fish, for example. And so it seems like potentially these things, take over some of those large predatory niches. That said, though, there are several examples of radiodonts that have survived into later parts of the Paleozoic. And there's even one that is from the Devonian period, from the early Devonian.
That's really cool. And did these arthropods shed their exoskeletons?
Yeah, so we assume that they would have shed their exoskeletons like all modern arthropods, as well as arthropod adjacent organisms. So arthropods belong to this large group of animals called the ecdysozoans, which are all characterized by molting. There's some sort of external cuticle. So that includes things like modern nematode worms as well, for example. So far, we have sort of indirect evidence of molting in radiodonts. And so some of the photos that I showed earlier of Cambropaster, you see this very stereotypical assemblage type where we have the head carapace and then the appendages are sort of displaced back behind the carapace. And sometimes we find some of the other body parts sort of displaced around that, but they're all sort of staying together in this one area. And so we're hypothesizing that those things might represent molt remains of Cambropaster, and that in fact some of these assemblages that we find with dozens of the specimens covering the bedding planes might represent mass molt assemblages where these animals are congregating together to shed their exoskeletons together. And we see similar behaviors in some modern arthropods. And they can do that for example, for mating or for defensive purposes.
So I guess a similar kind of follow-up question was, like modern arthropods, would these have larvae, or would they have kind of like instars, just like smaller versions of the adults? Is there any evidence either way?
So that's something that I'm really interested in. So far, the smallest published radiodont specimen is about 2 centimeters long. And so we don't have the early developmental stages that may have existed of these animals. I'm actually working on a little bit of material right now that will decrease the minimum size that we know of radiodonts. But so far, we haven't found anything that looks like a larval form yet. Now, that said, though, we do know of some other early arthropods called megachirons, or great appendage arthropods, more colloquially. And these guys, there have been some larvae that have been found from deposits in China. And so it's possible that radiodonts had a larval form that we just haven't found yet.
That's really cool. So speaking of size, so the things in the Burgess Shale, like even the biggest ones are only kind of a foot long or maybe even slightly larger than that, as far as we know. Was there something that was restraining their size, or was this just a small ecosystem, a small bodied ecosystem?
That's a difficult question to answer. I think, maybe one of the simplest explanations is that all of these major groups of animals had just evolved relatively, a relatively small interval of time before the deposits of the Burgess Shale. And so maybe we just hadn't gotten that sort of tail of the size distribution evolving yet. That said, though, some of these early arthropods, like radiodonts, are some of the largest known arthropods that we know. So I guess there are also some size constraints that control arthropod body size.
Right.
Like surface area to volume ratios that impact their ability to transfer gases with their environment, because these animals rely on passing oxygen, for example, through gill surfaces on their external body surface. Their size has to be highly related to their external body surface area.
Are you planning field work this summer?
We are, yes. We're hoping to go out in August for a month. And we're going to go back to that site that I showed where we were camping on the snow patch and try to bring a larger crew to that area and do a more extensive dig there.
Pretty exciting. Were you able to do field work last year?
No, so we actually haven't been out since 2018. So we are really excited to get back again.
Nice. Yeah, as I said, it looks like a beautiful, beautiful place to work. I'm a little bit jealous.
I think, it's not for everyone camping for 4 to 6 weeks in a remote area, but for me, it's like a vacation.
Yeah. All right, so I don't see any other questions. So I would like to thank Joe again for taking the time to be our virtual speaker for the month of April. This video will be posted probably in the next couple of days on our website under the virtual speaker series. And if you want to contact Joe about anything you've heard, he does have a website. So if you Google him, there's lots of ways to get a hold of him. I don't know if you have anything to add to that, Joe.
Yeah, you can also find me on Twitter. It's at Cambrojo.
All right, well, thanks again. And we will be posting about our next virtual speaker series for May in the coming week.
Thanks so much for having me.
Yes. So thanks again for everyone who was watching, and we will hopefully see you again next month.